Good afternoon, all of you. A very warm welcome to the Medicinal Biochemistry workshop. I hope you're all doing fine. I'm fine, Siksha. I'm also fine. Um, so, there was some timing problem was there. Um, so there was a vaccine workshop from 1 to 3 p.m. and then I need to give some gap time for such student could be relaxed. And then our next 3 to 5 will be our medicinal biochemistry. So there are around 17 to 18 students in this workshop. So it's 15:18 now. So let's wait for another 5 minutes and then we start. So around, let's say, 15:25 we start then. Okay. So, hi, my name is Dr. Simranjit Singh. I have been trainer with EduFabrica from last 1 year. Also was recently part as assistant professor at Kanpur and Rama University. Um, so I will start these lectures with little bit introduction about myself. So basically, I'm I had my education as a BTech in biotechnology in July 2009 from Shahiduddin Singh College of Engineering and Technology in Punjab Technical University, India. And then I went for my masters at the in Gießen, Justus Liebig University for agrobiotechnology. But I was part of a transgenic project. I left that in between, but I switched to then human medicine at the Max Planck Institute of Lung and Research in Bad Nauheim, where I worked under the Professor Offermans for the identification of novel endothelial specific molecules involved in the regulations of the tumor cell extravasation. Right, and then I got a doctoral position in April 2013, yeah, somewhere, and at the University of Göttingen, a place of Naturwissenschaft, that is of the scientist, in the Institute of Pharmacology and Toxicology. And the PhD was the topic under the molecular medicine, and the thesis title was redox regulation of PP1 and ES test regulation of CTGF in the cardiomyocytes. With respect to that, some papers were also published, but then I wished to come India for some personal reasons, and I joined as an assistant professor at Mata Gujari College in Fatehgarh Sahib. And after finishing that, I also joined EduFabrica, where I brought many subjects like MBBT, biochemistry, proteomics, and so on. And also was part of Rama University since last November. But recently, due to COVID situation, they just the faculty of 300 numbers were removed. So I'm also one of them. So yeah, that's something with my recent updates. Who am I? Um, so second thing is our lectures will be the timings of the lectures will be as I suggested in the last message. It got changed again and again and again. First it was 7 to I want to do it in a single day, but it was suggested to make it in 3 days. Then I suggested 2 to 4 a.m. or p.m. Sorry, but it was not working out. So it was a class of your vaccine from 1 to 3. So now final timings are around from 3:15 we will start. So around 3:15 we will start today, tomorrow, and Sunday. So it will be a 3 days workshop, 2 hours each day. Yeah. 2 hours each day, Friday, Saturday, and Sunday. And the link will be given to you just 5 minutes before, and then we can wait for 10 minutes or so, 5 minutes maybe. Then we'll kick out our sessions. So the objectives, as you might have seen it already. Um, is it this one or the other one? Yeah, so we will. Just a second. It's not. It should not be like this. Yeah, something I would like to take these paths within these 3 days. We will try to do maximum, like nucleic acid, then basics of your little bit about your biochemistry, then enzyme, lipids, amino acids, some virtual labs related to that. So we try to cover all these parts in our upcoming days, right? So actually this has to be here. Cool. So timing is clear, and the medium of our presentations, these workshops will be YouTube, online YouTube. So the lectures that we are delivering, they will be recorded under the name of Medicinal Biochemistry Workshop, June batch. So we will have 3 classes of 2 hours. So you can come to this YouTube channel anytime. So you can find them, these recorded versions, always with you. And please do subscribe to the channel. It will really help us, appreciate that, to go move forward and to make more nice live streaming and more videos for you. um yeah, so that was something introductory part. And what kind of communication would you like, students? Will it be English, or it's in English and Hindi both? So please tell me in the comments the language, medium of language, how it should be. So we continue with that then. Is it English, Hindi, or English, Hindi both? English, okay. If somebody, single student says English, so we will go with that, because it will make sense to we have common language for everyone then, right? Okay, so need to clear all the basics of our workshop. um, yeah, if you have any questions during the workshop or anything that you want to ask, so type me in the chat box, so I will come, I'm back after the chatting, and try to answer them. Yeah, so I'm not using Google Meets or Zoom because of the recording problem, so I have to record them and send this to your WhatsApp, and WhatsApp has a 64 MB limit, so I cannot. And these videos are mostly around 100 MBs, so there's a lot of chaos in these online platforms. So YouTube streaming is the best streaming for online classes, where I can deliver my lectures. And if you have anything you want to communicate, please write me in the chat box, so I will come back to answer. So next is, give me your introduction, students. Tell me your name, course, year, place, university, your hobbies, and what do you, where actually, where do you see yourself in upcoming 5 years? So each student answer them, and then we continue further. Yes, everyone. MSc Biotech 2020. Shivika Hundo. Think Vikram Jagdish Jhakar. Abu Nasser BMLT 2020. Siksha Swaroop. Completed Masters in Advanced Biochemistry from University of Madras. Oh, CSI. All the best. Suranjit Das. Okay. Bangalore. Oh, with big business empire. Shruti Kumari, MSc Medical Bio. Are you from Sydney? Okay, nice. Akshay Kumar, Physiotherapy, writing Hindi, nice. Mm-hmm. Maxofacial surgeon, wow. Tracker photography. Good, good. Kartike Parashar, MBBC final year. Oh, MPMCU reading. Mm-hmm. Good, good. Most welcome everyone to the part of medicinal biochemistry. So that was the just to get to know from each other, each perspective, where we are, who we are, and the how the sessions will go. So I will show all the lectures here in this field. Oh, there's one more. Sai Sovamya, Integral MSc Biotechnology, third year, Vishakhapatnam, Andhra Pradesh, Geetam University, Vizag. Yeah, great. Most welcome. Everyone here, each participant, you're most welcome to the class. Um, So as I said, in this medicinal biochemistry, we will deal with the nucleic acid first here to understand about structure of DNA, all the RNA, and how the transcription and how their functioning is. So we will deal with that part only in this today's lecture. So first thing first, structure of DNA. So in this, we will understand about the concept of DNA, structures, packaging in the nucleus, demonstrate the concept of central dogma in the molecular biology. So from the history perspective, nucleic acid was first isolated by Friedrich Miescher in 1869. Then Friedrich Griffith discovered that factor in disease bacteria that can transform harmless bacteria into deadly bacteria. Then because of his transformation experiment, he found it in that part. Then Rosalind Franklin, he got the X-ray of our DNA in 1952. Then Watson and Crick, they are the first one who discovered the DNA molecule from the Franklin X-ray itself. So they describe each part in more details about it. And then the Chargaff rule came as a purines and pyrimidines. That purines are in equal amount of pyrimidines, right? So this is the DNA structure, which we can see the backbone is made up of sugar and phosphate. In the center, we having nitrogenous base pair. So that is your purines, uh, adenine and guanine, and your pyrimidines are thymine and cytosine, right? So adenine and thymine, they are making disulfide bridges, whereas guanine and cytosine, they are making, hydrogen bonds, triple hydrogen bond, and here double hydrogen bond. So DNA consists of double helix with two chains having this sugar phosphate outside and your nitrogenous base pair inside, and is made up of millions tons of subunits of nucleotides. And prokaryotes are nucleoid, and plasmids where the eukaryotes are present in the chromatin of the nucleus. So this DNA has mainly three main structures, that is deoxyribose, base pair, and phosphate. So this is the deoxyribose sugar, that is your pentose sugar. To that your phosphate group is attached, and also a nitrogenous group, nitrogenous base group here. So in this way, that makes a component of our DNA structure. And this sugar could be of two types, ribose sugar and deoxyribose sugar. So we start to count them 1' and 2' and 3' and 4' and 5', right? So ribose sugar here in the second prime end will have your hydroxyl group. On the other side, the second prime end will have no hydroxyl group. So this is deoxyribose sugar, and this will be your ribose sugar. Yeah. So in this way, 1, 2, 3, 4, 5 carbon chain amino acids. Your sugar bonds are made. And deoxyribose is present in DNA, whereas ribose is present in your RNA. And the bases, so they are divided into two groups, nitrogenous base pairs, pyrimidines and purines. The purines, that is your AG, they are actually 5 and 6 membered ring, adenine and guanine, whereas pyrimidines, they are thymine and cytosine. There are 6 membered rings, so 1, 2, 3, 4, 5, 6. So they eventually, what will happen, this nitrogenous base pair will get attached to the sugar group, and the sugar group will attach to the phosphate group. So on the side chain, they will make phosphate, sugar, and each sugar will keep joining the respective nitrogenous base pair, like A, T, 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 C, C, C. And correspondingly, there will be their respective purines and pyrimidines are getting attached. And remember, each number of purines will be always be equal amount of your pyrimidines. They will be never be in different ratio. That is your Chargaff rule is stating that. And further, there are nucleosides and nucleotides. Nucleosides are the one which has only deoxyribose sugar and nitrogenous base pair, whereas the nucleotides are the one which has deoxyribonucleosides plus phosphate group attached to it. That is a phosphoric acid. So when phosphoric group is attached, then it's nucleotides. If phosphate group is not attached, then it's the nucleosides. So we're having these four nitrogenous base pairs: adenine, guanine, cytosine, and thymine, right? So adenine will attach to deoxyadenosine. Uh, then guanine, if there's a base and deoxyribose, then it will be deoxyguanosine. In the same case, if we add the phosphate group, the name would be then deoxyadenosine monophosphate. And we will abbreviate them as DMP, DGMP, DCMP, and DTMPs. And the nucleotide structure, as we have seen, it is made up of your phosphate, sugar, and nitrogenous base pair. And these groups are attached by the phosphodiester linkage. Yeah, so these sugar and phosphate, they are attached by the phosphodiester linkage, and the sugar and the nitrogenous base, they are attached by the glycosidic linkage. This is your glycosidic linkage, and so on. So this DNA and hydrogen bonding, how they are correlated. So if some question comes that why DNA has a double helix structure, the reason behind the double helix structure of DNA is that it has these hydrogen bonds between two nitrogenous base pair. That's the thing which keeps this structure in this double helix structure of DNA. So they are made up of two strands, as we know, nucleotides that are joined by the hydrogen bonds, and they make this complementary base pairing between your adenine and thymine and cytosine and guanine, and each these pairs are connected by the hydrogen bonds. And always remember, one pyrimidine will be equivalent to the one purine during the whole course of your DNA, and that's why your DNA is double helix structure. Then there's anti-parallel strands, which starts, so we having this sugar 1, 2, 3, 4 and 5. So on the fifth group, your phosphate groups are attached mostly. On the other side, we are having this 3' end uh, carbon end, and here also 3' end, and we have also 5' end here. So this goes from this direction, 5' to 3', 5' to 3'. And in between, these are our nitrogenous base pair, adenine and thymine. They are making the disulfide bridges, and this guanine and cytosine, they are making the triple bond. So the strand runs opposite in each direction, and the 5' always have this phosphate group attached to it. Then there is a Watson and Crick model. So in this model, so far it's a Watson and Crick model. So what we have to discuss here is the 5' end of your sugar will be attached to the phosphate group here, and the 3' end of other side will be attached to the sugar group. So together these two bonds, they will make a phosphodiester bonds. Yeah, and then so they are anti-parallel to each other. Hydrogen base pairs are there. Sugar and phosphate is outside, base is inside, and beyond that, the coiling of DNA is right-handed. That is, this is the form of bDNA. So there are three types of DNA we will discuss soon. And the one complete turns includes 34 angstrom. So this, what does it mean by one complete turn? This complete turn includes your major groove and minor groove. And this within one major groove and minor groove, you have around 10 mononucleotides. You have 10 mononucleotides. So here we can see this is the length of one turn, 3.4 nanometer, and this is the major groove here, the deep, narrow, and this is the deep and broad, and this is narrow, thin. This is the size of 1.2, this is 2.2, together 3.4. So this includes your major groove and minor groove, and within this group you have around 10 nucleotides. So you have 10 nucleotides in between. And the distance between each nucleotide is known as rise. Rise per base turn is called as rise. And you can see there is some angle shift. So there is some, if you just make them go here and this one there, so they are making some angle here, these nucleotides. So this angle is called as tilt. And that's these distances, and the diameter of this DNA is around 2 nanometer. And this is ribbon model, this is space filling model. So yeah, so there are three types of DNA, that is aDNA, bDNA and zDNA. So the right now the model that we are seeing is the bDNA, which is the most common one. And on the right hand side is the same model showing in the space filling model with these circular balls here. Yeah. So in summary, what we have discussed that in the DNA Watson and Crick model, there are polynucleotide chains coiled together to form right-handed helix. They are anti-parallel, both runs from 5' to 3' end in opposite direction. And nitrogenous base pairs are there to one another to form hydrogen bonds. And the base pair stacks on each other located inside the helix. And each complete turn of DNA is around 3.4 nanometer long and which contains 10 bases. And each turn along the molecule contains your one major groove and one minor groove. So that's the summary of what we have just discussed here all together. Now comes the three types of DNA are present in all, that is aDNA, bDNA and zDNA. So aDNA and bDNA, they are bothly right-handed helix, and bDNA is mostly found in the living system, whereas the aDNA has 11 base pair per turn and the bDNA has 10 base pairs per turn. And the diameter here is 2.3 nanometer. On the other side, the bDNA diameter is around 2.0 nanometer. So this is a bit, yeah. Further, the zDNA is the left-handed helix, and zDNA is called because it's given Z kind of structure, not the helix structure. And here we have more base pairs here, 12 base pairs are there, that's why it's elongated, and the diameter is a bit less, that is 1.8 nanometer, so it will be slim. So here we can see they are a bit slim, right? And this is a bit broader, and this is, oops, this is intermediate. This one has 10 nucleotide base pairs, this has 12 per turn, and this has 11. And this gives a Z-shaped structure, this zDNA, and these B's are helix. This is a right-handed helix, and this is left-handed helix. Yeah. Yeah. So let's discuss each of these forms. Oh yeah, one more. So I was discussing last time the major groove and minor groove. So in bDNA, we can see the major groove is quite deep, but the minor groove is not that deep here. In the aDNA, it's opposite. Minor groove is deep, major groove is not. So that's how the difference between A and bDNA. And there is a tilt, as I was discussing, the angle between two nucleotide pairs, and there is a thing called rise, distance between two nitrogenous base pair. So this is overall proportion, this is helix rotation sense. These are the major things we will be discussing now. Major groove, minor groove, yeah, this one also. So overall proportion, the aDNA is short and broad, this is longer and thinner, and this is elongated and slim, right? And the rise, the distance between each nitrogenous base pair, it will be around 2.3 angstrom here, and this will be 3.32, and this is the largest 3.8. That's why it's the elongated, this is longer, and this is the shortest. And the diameter of this A is the highest, that's why it's broad. This is intermediate, that's why it's thinner, and this is the least, that is slim. Further, helix rotation is right-handed here, right-handed, and this is left-handed. It has 12 base pairs per turn, this has 10, and this has 11, right? And so the angle between two nucleotide base pairs in the aDNA is quite high, plus 19, this is minus 1.2, and this is minus 9 degrees Celsius. Further, the major groove here is extremely narrow but very deep. This is wide with deep intermediate depth. This is flattened here in this case. And the minor groove here is very broad and shallow, and this is narrow with intermediate depth. And this is extremely narrow but very deep. So that's why the structure of ZDNA is coming in this way, because of the major and minor groove. Also the angles are there, and we have a number of more nitrogenous base pair. So that's the major difference between your A, B and Z DNA. And there are also these DNAs could be classified as a linear DNA and circular DNA. The linear DNA are the one in which the ends are free, and they are mostly present in the eukaryotic cells. Where the circular DNA, which does not have free ends, they are present in the nucleoid regions of prokaryotic cells, and they are always in circular form. So some example of circular DNA and end free linear DNA. So write down these answers, answer them first 5 questions, then we can go for further 5 later. So DNA stands for, what is the shape of DNA? Who established the structure of DNA? Adenine always pairs with, and the sides of DNA ladder are deoxyribose and which one? So answer these questions into the chat box. Answer them, students. Answer the first 5 questions. Then we go for the next 5. I know they are the basics, but it's good to know. Some of you might give the wrong answers also. Yeah, nice, nice. Answer all of 5, students. Write down 1, 2, 3, 4, 5 like this. Not single. I will not get to know which answer you are doing. Okay, I will answer them. This is deoxyribonucleic acid. The shape of DNA is double stranded helix, and this was established by Watson and Crick. And adenine pairs with guanine, and the sides of the DNA ladders are deoxyribose and phosphate group. Yeah, so now answer the 6, 7, 8, 9, 10. Answer them like 6. Write down in the chat box 6, then answer, then 7, then answer, then 8, then answer 9 and 10. Answer them together with the numbers. So I will keep it up. So answer these 5 questions. Yes, students? Okay, I will answer them. Whosoever, how many students have answered? Only 2 students have answered. What with the others? There are 40 students in the class. So, oh, there are 15 actually. So 15 doesn't know the answers, or they're just here sitting in their bed, lying down with the phone open and just listening to me. Or there are some serious who want to get up, sit in the chair and be serious about it. Okay, deoxyribonucleic acid, double helix structure. This is the previous Vivek Pancholi. I'm asking from 6, 7, 8 now. Adenine, okay, hydrogen bond, nucleotide, adenine, yeah, right. Adrika Maji, you got the right answers. Okay. Rest, I think they are not interested in this answering. They know very well. Okay, so we are done till here. So we have learned the basics of structure of DNA as a genetic material. We could correlate these DNA structure with replication, transcription, and translation in our upcoming lectures. Yeah, that's a reference. So okay. So these lectures will be shared as soon as I'm finished with them. So I will share with you. So it's not the basics of molecular biology, but I want to continue with the RNA now. So we had discussed, we had a vision about DNA so far. I want to discuss now RNA. That is RNA, which is a biological important type of molecule, consists of long chain of nucleotide units, and each nucleotide here consists of nitrogenous base pair, that is your ribose sugar and phosphate group. So you have a single chain amino acid, and then there's a double, then again single, then again single. So in this way. So it's just the main difference is that it has deoxyribose sugar, ribose sugar in this RNA. And sometimes this RNA could be double stranded. It's mainly in the viruses, double stranded RNA viruses it could happen. So they are basically, from the function point of view, there are three types of RNA: messenger RNA, ribosomal RNA, and transfer RNA. So messenger RNA, they codes for proteins for all organisms, and ribosomal for translation, and transfer RNA for also translation. Whereas in the post-transcriptional modification, it could be snRNA, yRNA, telomerase RNA, which has the function as splicing, RNA processing, and telomere synthesis respectively. And there is antisense RNA, that is aRNA, which has the functions of various translation block, transcriptional attenuations, mRNA stabilization. Further, your messenger RNA, which act as a postman, which take the message from DNA and go out of the nucleus and go into the cytoplasm, and with the help of ribosomes and tRNA, they translate the message of the DNA into the form of proteins. So proteins are made by combinations of polypeptide chain of your amino acids, and they get folded into the active form in order to function. So that's basically the function of your transcription and translation plays with the help of your DNA, RNA, and protein, and this is known as central dogma of life. So mRNA, which is carrying information from protein sequence to ribosomes, the protein synthesis factories in the cell. So it is coded so that every 3 nucleotide correspond to 1 amino acids. And in eukaryotic cells, one precursor's mRNA has been transcribed from the DNA, and it is processed to mature mRNA, and this removes its intron. That is non-coding sections of the pre-mRNA. So you have a coding strand and you have a non-coding strand. The coding strand will be your CAATTCGC. So like in the previous case, we were discussing about DNA replication. So respect to other, it was producing A was producing T was producing A. But in the case of mRNA, it will be uracil will be coming out, not the thymine. So that's the basic difference. Then mRNA is exported from nucleus to cytoplasm, where it is bound to the ribosomes and translated into the corresponding protein with the help of tRNA. So this statement that I have just said that before, that your mRNA is exported from the nucleus to cytoplasm, where it is bound to ribosomes and translated into the corresponding protein with the help of tRNA. Just a second, students, I'm getting a lot of disturbance from outside. Let me see. Just coming back. Some construction is happening, so we can't stop that. So yeah, this statement that mRNA is exported from nucleus to cytoplasm, where it is bound to ribosomes and translated into its corresponding protein form with the help of tRNA. So here we can see that this statement, let's discuss this thing first. that what are DNA genes from the perspective from the animation. So we are all human beings are different from each other just because of the ATCG. So they are different from each other, and that's why we are different. Sorry, I have to remove my mic.
For building all living things. The instructions for building all living things, whether a fish, a bacterium, a tree, an insect, or a person, are found in a molecule called DNA. DNA is made up of 4 tiny building blocks. Each building block has a backbone and a chemical base that is represented by the letter A, C, G, or T. DNA molecules have 2 strands that are joined together through complementary base pairing. A pairs with T, and C pairs with G. These building blocks function together in larger units called genes. Each gene is a set of instructions for building a specific protein. A complete set of genes is called a genome. A genome is a set of instructions for building an entire organism. Every person's genome has the same genes arranged in the same order. But small differences in the sequence of the bases in our genes make each person unique.
That's why we are different from each other. So whatever I will discuss will be sharing over the group. So next is protein. What are proteins are?
Proteins carry out most of the body's functions, and they make up the bulk of its structure, from hair and fingernails to bone and muscle. In fact, nearly everything in our bodies is made from or by proteins. The instructions for building proteins are found in segments of DNA called genes. Proteins are made of long chains of building blocks called amino acids. There are 20 amino acids in all. Put together in different combinations, amino acids form proteins with a broad variety of shapes. This diversity of form gives proteins their many functions. The proteins a cell makes determine what it can do. Our cells are constantly at work, producing the thousands of different proteins that keep our bodies functioning.
That was how the protein works in our body. Now we have different genes and they have different functions. What if we bring one gene to one organism to another organism, get the function? Like the jellyfish, they are producing, you know, um, fluorescent light in the deep ocean. So we get that gene, we put into the fish. So the fishes will start to glow. What if we take a spider web and spider, so they are producing silk web, and we took that gene into the yeast, and we can have a large production of silk web out of it. So that's the thing. The next animation will be discussing same gene, different organism.
These may look like ordinary fish. What you can't see is that they have a gene in them that codes for green fluorescent protein. But shine a blue light on it, and GFP emits a bright green glow. What might surprise you is that the gene for GFP came from a jellyfish. GFP was first discovered in jellyfish over 50 years ago. Since then, scientists have been using it to study all kinds of biological processes. What's amazing is that this gene from jellyfish can be put into just about any living thing: animals, fungi, plants, bacteria. They can read the DNA code of a gene and build GFP that is identical to the jellyfish protein. That same concept applies to tons of other genes too. The gene that spiders use to make silk webs, for example, can be put into yeast cells. Harvest the silk protein that the yeast cells make, and you've got the raw material to make a strong, lightweight fiber. In fact, a company is using this approach to bring yeast-grown spider silk fabrics to market. How is it then that fish and yeast can read DNA from a completely different organism? It's because, despite their differences, there's something fundamentally the same about how all living things read genes to build proteins.
Yeah. That I have just discussed with you. Now comes the main part, that is your transcription and translation. Let's start.
Transcription and translation are the processes that all living things use to read the information in DNA and build proteins. Each gene in a cell's DNA codes for a specific protein. During transcription, cells make a copy of a gene out of mRNA. During translation, they use that mRNA copy as a blueprint to build proteins out of amino acids. These proteins perform the tasks that make living things function. The flow of genetic information from DNA to mRNA to protein is central to life.
So let's choose with a gene. Let's say this one.
Inside the nucleus, RNA polymerase attaches to a gene. It separates the two complementary DNA strands, exposing the gene's unique sequence of bases.
So in this, C will pair with, in the mRNA with G, T will pair with A will pair with U, and U, C, G, and so on.
When the end of the gene is reached, transcription is complete. The new strand of mRNA passes out of the nucleus and into the cytoplasm, where translation takes place. The instructions in an mRNA strand are read in groups of 3 bases called codons. Each codon must be translated, one after another, in order to build a functioning new protein. The key structures in that process are the ribosome and transfer RNA, tRNA. Each tRNA molecule has its own anticodon and a matching amino acid, the building blocks of proteins. To begin translation, a tRNA molecule coded for the amino acid methionine docks with a ribosome. The ribosome attaches to the end of the mRNA strand. It slides along until it finds a start codon, AUG. Through complementary base pairing, the start codon pairs with the anticodon on the tRNA molecule, signaling the beginning of the protein instructions. The methionine molecule is now the first amino acid in the new protein. The ribosome shifts to read the next codon. Each codon is recognized by one of 20 different tRNA molecules. Each type of tRNA carries a different amino acid. The ribosome continues moving along the mRNA strand, one codon at a time, matching it to a complementary tRNA. It adds each amino acid to the growing protein in just the right order. The sequence of amino acids determines the final shape of the protein and the function that it will carry out. Give translation a try.
Yeah, so the transcription is done. Now the translation. So CGC will have GCG, arginine. The corresponding amino acids are attached to them. Then UUU, then it will be AAA, phenylalanine will join. On UAC, it will be AUG, tyrosine, and list goes on.
When the ribosome reaches a stop codon, it detaches from the mRNA strand. The chain of amino acids folds into its final form and the new protein is complete. This protein, aminoacyl tRNA synthetase, will now attach the right amino acid to the proper tRNA molecule, charging the tRNA so that it can participate in protein synthesis. Each type of amino acid has its own enzyme. The incredible processes of transcription and translation enable cells to carry out all of the functions necessary for life.
So that was in brief. I hope you will remember this: transcription and translation. So wanted to have some of these animations so that the things that we are discussing could be understood in better way. So let's continue. So this is transfer RNA, which is a small RNA, second type of RNA, around 18 nucleotides, which has ribosomal site synthesis during translation. From the top, it will join with amino acids by the ester bond. And then in the bottom, there's mRNA, and it's attached by the anticodones here. It's a clover leaf structure, and that site binds to a specific sequence on the messenger RNA chain through hydrogen bonding. And this third type is your ribosomal RNA, which is a catalytic component of the ribosomes. They are of two types, 70 and 80. 70 is made up of 50 and 30S, and 80 is made up of 60 and 40S. And the main difference between RNA and DNA, as we know, it's a uracil and thymine, and of the sugar, ribose and deoxyribose sugar, and RNA is mostly single stranded and DNA is a double stranded. Now comes your protein, which is a polypeptide chain of amino acids around this globular form, and they are made up of basically 20 amino acids. So they make this structure together. These cysteines are making disulfide bridges together. They get the quaternary structure, and they could be present in peanuts, red kidney beans, pulses, fish, white chickpea, your eggs, red meat, chicken breast, all these things they are present. So basic players that are playing in this biochemistry here are your DNA, RNA, and proteins, which are the central dogma of life. So DNA could replicate themselves, then transcription could happen, and then translation. So in the DNA replication, what is happening first? So in the DNA replication, that's the basis for biological inheritance, which is a fundamental process occurring in all living organisms. So basically, it's a semi-conservative nature in which the parent DNA gives its half part, 50% to the offsprings. So here in this DNA replication, we can see that there are various enzymes are playing a major role here. That is topoisomerase, which will open up the DNA, and helicase will separate the nucleotides. Then there will be single-stranded binding proteins which will attach in order to stabilize the nucleotides. Then further, there will be two strands, leading and lagging strands. The leading strand will start producing the corresponding nucleotides with the help of DNA polymerase enzyme. On the other side, it will also do the same thing, but it will do in the fragments. So these fragments called as Okazaki fragments, and they could be healed by the DNA ligase enzyme. Also, to initiate this reaction, your DNA primase and RNA primase are helpful. In this way, we have a lagging strand, and this is the leading strand. And always the DNA replication always happen in the direction of 5' to 3' direction. So this is from 5' to 3' direction, and this one is also this 5 prime to 3 prime direction. Sorry, from here 5 prime to 3 prime direction on this direction. Then comes transcription, in which the equal amount of RNAs are produced, the copies of RNA in the transcription. It could also be reverse transcribed, and also instead of thymine, uracils are produced in them, and the enzyme that is helpful in during this process is your RNA polymerase, which produce your complementary antiparallel RNA strand. So the stretch of DNA, so here we can see we having our 3 prime to 5 prime and DNA. It will get unwinded up and you will have your RNA polymerase attached to it. Then RNA polymerase, what it will do, it will transcribe your DNA into the in this form, and then the corresponding messenger RNA will produce against it and will get separated, and we will have a separate single or this double chained RNA. Also during this process, RNA synthesis and processing will happen, where your chromosomal DNA like exon 1, 2 and 3, they will be removed. And these are the exon 1, 2, 3 are the coding regions, and there's introns which are non-coding regions. So they will get separated during the RNA splicing, and we will have mainly your exon 1, 2 and 3. And also there's a reverse transcription is happening during this process, in which they replicate the genomes by reverse transcribing the DNA copies from their RNA. So like for HIV, there's a RNA, HIV RNA, which will make reverse transcriptase and will have HIV DNA. So all in all, what we can say that these are the general cases that the DNA got self-copied, leading to production of RNA. Then RNA is leading to the production of your protein. And RNA could also self-replicate and could go back to the DNA. So this is a special case that could happen. And during the translation, it requires the ribosomes and tRNA. So ribosome, small unit, and large subunit, they come to the messenger RNA, and they first, like let's say methionine transfer RNA will come. It will attach, it will find AUG starting codon, and then further glycine will come, leucine, valine, alanine. They keep coming, and tRNA will keep removing, but they will keep making your amino acid join. That's what we have seen in that video very nicely. So this is the genetic code of our DNA. That is, what you do is you write UCAG on the left hand side, and then you write UCAG on the top, and then you write UCAG all 4 letters on the third side 4 times. So you will have 4 boxes at the end. So out of these 4 boxes, start writing the first base from here, second from here, and third base from here. So in this way, each box will have 4 amino acid sequence, right? And then start naming them from the start codon here, methionine. Then there is a stop codon, UAA, UAG, UGA, and beyond that, start naming from like full boxes like serine, leucine, proline, threonine, valine, alanine, and list goes on. And then you can start naming the 1R in parallel together, phenylalanine, leucine, and also so on. So that's it. We have discussed in detail about nucleic acids here so far, how the structure of nucleic acids looks like, what are the various types of nucleic acids are, how the transcription and translation works out. We have discussed them in very nice detail and with animation also. So on this point, we will take a 5, so it's 16:30. So we will take a 5 minutes break here. So we will see us at around 16:38 or 37 now. Yeah, and then we continue further to the respective course. Okay. Yeah, so genome we will not discuss. Yeah, some tools I would like to discuss as they are written in your virtual lab. So we have discussed in today's 2 hours lecture so far, that is your structure and properties of purines and pyrimidines, nucleosides, nucleotides, your structure of DNA and RNA, and the central dogma of the life. Now I will take you to the virtual lab of PCR, DNA isolation, and gel electrophoresis. Yeah, so these things will be covered now. First thing is PCR. So this will do later. So PCR is basically an extremely versatile technique for copying DNA. It allows single DNA copied to be used. So in order to explain this, I have another presentation. So uh, this is a technique in which we can have a lot of small sequence of DNA into million multiple copies. So a short history. So they have been discovered by Dr. Carey Mullis, who have developed the PCR, and the first publication was done at the CETUS Corporation, which appears in the Science. And in '86, then purified Taq polymerase was used in PCR. Then Perkin Elmer, they introduces automated thermocycler in 1988. Then in '89, science declares your Taq polymerase to be the molecule of the year. So in this process, mainly first denaturation has happened. Your double stranded DNA, it's got melted to into a single stranded DNA. At 92 degrees Celsius, then the temperature is decreased, where your forward and reverse primer, they got attached. At 50 to 70 degrees Celsius, then they bring to the 72 degrees Celsius, where your DNA polymerase will bind to the anneal primers like Taq polymerase and will start producing the corresponding nucleotides. So at the end, you will have a single same copy of your DNA standards. So by the end of a PCR 35th cycle, like we were discussing in this part, so you will have a first cycle, you will have 4 copies, so it will go 2 raised to power n way. So 2 raised to power 2, 2 raised to power 3, so 8 copies, 16 copies, 32 copies. So by the 35th cycle, you will have around 68 billion copies. So they will amplify in the exponential way. And the components that are required during the PCR are magnesium chloride, buffer, DNTPs, primers, DNA polymerase, and target DNA. So basically we need a basic sequence you want to target, and we need primers of 20 to 30 base pairs. Then thermal cycler is required in order to make it happen. Then Taq polymerase is required, which is not inactivated. And the various applications could be from genetic engineering, sequencing, and molecular identification. The instrumentation that we use in this case are these kind of thermocyclers. This is 4-in-1 thermocycler. This is the oldest one. This is mostly in the labs are, and this is the latest high-tech light cycler thermocycler. So that was the briefing about PCR. So how PCR looks like, how PCR works like. So we'll share this presentation of PCR, also the introductory part. this digit came. Yeah. So we will now go for a virtual lab now. This will be our last chapter, the gel electrophorese in DNA isolation. We will do it tomorrow. Yeah, not today. So this one is the best.
Before beginning this protocol, students must fill out the worksheet provided on the last page of this document. This will allow students to calculate the appropriate volume of reagents to mix in the creation of a master cocktail. The purpose of a cocktail is to mix all the common reagents in a multi-sample procedure to avoid excess pipetting and potential errors. In this example of Protocol 2A, 4 students are amplifying the actinin 3 gene from all 4 of their DNA samples. This information is filled out on the left-hand side of the worksheet. On the right-hand side, students use the equations to determine the number of tubes needed and the multiplier for reagent volumes. In addition to the 4 DNA samples, a negative control sample is always included. Therefore, 5 tubes are needed for the PCR reaction. In the creation of the cocktail, pipetting error is accounted for by adding another sample. Therefore, the multiplier for this group is 6. Each row of Table 1 represents a common reagent used in all the samples undergoing a PCR reaction. The multiplier is filled out in all rows of the multiplier column of Table 1. Moving across each row, the volume indicated per reaction is multiplied by the multiplier. .5 times 6 equals 75 microliters of red tack added to the cocktail. 11.5 times 6 equals 69 microliters of water added to the cocktail. 0.5 times 6 equals 3 microliters of each of the forward and reverse primer added to the cocktail. Finally, the sum of these volumes should be equal to the product of the final row. 75 plus 69 plus 3 plus 3 equals 150 microliters of total volume in the cocktail. This amount matches 25 times 6. Table 2 is used as a guide for labeling of the PCR samples. As indicated by the equations, this group will be using 5 tubes. The class must collectively number the PCR samples to not confuse samples amongst the groups. In this example, these students have been assigned samples numbered 5 through 9. The sample names are filled out in the second column, and the primers used for each sample are filled out in the last 2 columns. In this example, the forward primer is actinin 3F, and the reverse primer is actinin 3R. The same primers are used for all samples. The common reagents provided are molecular biology water,
a forward primer, which you should be sure to check for the marking of PCR to ensure that you are using the appropriate concentration. A reverse primer, and red tack ready mix. The red tack will need to be thawed prior to use, but once thawed, keep the solution on ice to prevent enzyme degradation. Next, prepare the cocktail by using the volumes and reagents indicated on the worksheet. Obtain a new tube and label it as the cocktail. When creating a cocktail, it is best to start with the largest volumes. Though red tack is the largest volume, the solution should be kept on ice as long as possible, so instead the water is added first. When adding each of the reagents, be sure to use the appropriate micropipetter for the volume being added. Next, the primers are added. It is best to pipette small volumes directly into larger volumes to avoid loss of the reagent. Finally, add the red tack and immediately return the stock to ice. Mix the solution and centrifuge all cocktail tubes created by all groups in the class together. Press and hold the short button to do a brief spin.
Just a second.
The collected cocktail solution will be split amongst the 5 tubes in this example. PCR tubes are provided in strips of 4, so if you need 5 tubes, use 2 strips of tubes. In this case, to amplify actinin 3, 24 microliters of cocktail solution is added to each of the 5 tubes. Before adding DNA to the PCR reactions, make sure that the tubes are labeled. Make sure all DNA samples are mixed before transferring the solution. One microliter is a very small volume, but you should still be able to see a drop in the tip. Put the tip all the way into the bottom of the tube and pump up and down on the plunger before expelling to the second stop. This helps to ensure that the 1 microliter of DNA has been expelled into the reaction. Cap the tubes tightly and flick to mix the samples. Use the mini microcentrifuge with the 8-tube strip head to collect the solution at the bottom of the tube. It is good practice to always mix and collect solutions before every step, especially if the solutions have been sitting for a while. Turn the thermocycler on with the switch in the back. Place the tubes in the thermocycler by pressing the metal latch and lifting up on the lid. Tubes can be placed in any spot or orientation. Close the lid and make sure the metal latch has clicked into place. Heat cycling programs have already been programmed into the thermocycler, so all you need to do is run them by pressing the Proceed button. Use the horizontal arrows to navigate to the program of interest, in this case ACTN, to amplify the actinin-3 locus, and hit Proceed. The heated lid should be enabled by hitting Proceed to prevent extreme condensation at the top of the tube. The thermocycler will ramp to the initial temperature and begin timing the incubation. It is good practice to make sure the machine reaches this step with no errors. After it has initiated properly, the machine can be left while the reaction proceeds for several hours. Once the run is complete, the cycler can be turned off by simply flipping the switch in the back of the machine.
Okay, so this PCR is over. Okay. Okay. So this is the today's lectures. So I've shared all the content so far. So we will see us students tomorrow the same time around 3 o'clock, 3:10, and then we continue till this here. So anyone has any questions so far can ask now. I can discuss it and we can make some discussions here. The questions related to this topic, not something other than that. Okay, if not, then we see us tomorrow at 3 p.m. And we continue further with the DNA isolation. So thank you very much and see you tomorrow. Bye bye. Take care.